I had a way then, losing it all on my own.
First, the onion. Secondly, the microscope. Then the glass slides. Cotton swabs. The cover slip. A Bunsen burner that we didn't use. Lugol that is a toxic stains. Methylene blue, non-toxic. And alcohol. Well, the first lab was divided into parts. The first procedure was the onion, and then we used the cheek for the saliva. Well, the procedure of the onion starts with getting 4 of the glass slides and cover slips and make sure that we have to wash it and dry it. Then remove 2 epidermal cells from the onion and leave it 30 seconds on the alcohol. After that, we have to remove both of the epidermal cells from the alcohol and put one in the lugol and the other one in the blue epithelium. Then remove them from the stainers and wash the tissues for 30 seconds. Finally, put the cover slips on the microscope and make sure that there are no bubbles on the cover slip. Well, the second procedure, we couldn't do it completely, but it's about that 2 of the group will offer their cheek tissue. Then we use cotton swabs for removing epithelium from the cheek. Then place the content from the cheek on the microscope slide and wait until it dries completely. Then the part that we couldn't do is that go with you, Parra, and fix the tissue with the Bunsen burner. Finally, apply a drop of Lugol and methylene blue and wait 2 minutes. Then wash it. Do not apply water directly. Just let the water run over the slide very gently. Well, the purpose of this first laboratory is to study the structure of the onion and the cheek epidermal cells with the Lugol and blue methylene. Well, the truth is that I didn't take these pictures from my camera. I took them from Google. This is how we can see the epidermal cells in Lugol. These are epidermal cells in blue epithelium. It looks like bricks, but the truth is that these dots are the nucleolus inside the nucleus. Is the control center or brain of the cell. It stores all the genetic information of the organism. The nucleolus' primary function is to assemble the ribosomes in the nucleus. Well, the thing is like in flowering plants like the onion, it contains both male and female parts. So I can say that this cell uses a division like mitosis because this make that at the same time there is a meiosis using his own sex cells. It is like a division in which it reproduces alone, like in mitosis. I think that the onion uses asexual reproduction. Well, the materials that we used were a microscope, a glass with water from the floor after a rain, and a pipette. Well, the process in which this second lab was divided in two parts. The first part is to take the water with the pipette and put it on the glass slides. Then the glass slides are in the microscope, so from there we can see if there is life in the water that we took. Some parts or sometimes we saw that the water was really contaminated, so there was no life in the water. But sometimes we saw parasites. Also, we saw rotifers, um, barsurias, parasitic protozoa, like fighting or finding his food in the water. These things or these animals were really big. This is a rotifer inside the microscope. It was really big. The first thing that I saw was that the rotifers were moving so slow. They were moving side to side, like trying to find something. But the parasites were too fast, like worms, like trying to escape from the water. Well, the importance of microorganisms in the environment are that they help us in the decomposition of the species in the environment. They also produce oxygen and cause evolution in the other species. If the microorganisms disappear, there will be less oxygen and there will not be like an advance in the evolution.